great pleasure to be speaking here today. I would like to thank the organizers, Tiana, Marinka, Natasha, and other co-organizers for giving me this opportunity to present my work. Today, I will be speaking about a network perspective on mutagenic processes in cancer.
Okay. It might take a few more minutes to try
this is
to
a
set
subject
up
that
the
my group
audio
and
for
I've
the
been
speaker here in the
working
room.
on for a couple of years already. And today I would like to focus on some of the approaches that we developed that involved network biology. I will start with a brief introduction to mutational processes in cancer. So as we know, a cell, each cell is subject, DNA in every single cell is subject to a number of processes that lead to DNA damage. This may be environmental processes such as UV light or sun. It may be also processes that are related to a regular functioning of the cell that are error prone. Those processes lead to about 10 to the 5 lesions per cell per day. Fortunately, they are very efficiently repaired by DNA repair mechanisms. And thanks to this DNA repair mechanism, we only have a very limited number of unrepaired lesions that turn to be mutations. One of the important observations related to mutations that are caused by those either environmental processes or cellular processes is that they often specific mutagenic processes lead to specific mutational patterns. For example, UV light gives characteristic mutation pattern where C is mutated to T in a condition-specific way. Specifically, if C is preceded by T, then we have mutation then C to T mutation happens more frequently. UV light is not the only mutagen that has this very characteristic mutation pattern. And it's very interesting to be able to investigate those patterns and link them to. In order to do so, we need to have some formal definition and representation of those patterns, and this is provided by so-called mutational signatures. And the one method that was proposed by Alexander et al. That is broadly used is to consider various partition all the mutations into mutation categories. Their method suggested to, and the approach that is now being picked up by most of the studies, is to divide all the mutational mutation into 96 category based on the triples of nucleotides, but the mutated nucleotides is the central one, and the two flanking nucleotide provides the counters. In this way, we have all this mutation of central nucleotides, and what you probably cannot see here are those all these triples that provide the context, and then the mutational signatures or mutation as a probability distribution over all these mutation categories. So given this definition, there's some examples of other mutational signatures. The top here is mutational signature of ApoB, then signatures that was associated with smoking, then yet another one with cancer drugs. So of course, we don't know all the mutagens yet. However, we can ask the question, what are the some minimal or some optimal number of signatures that can explain the mutational pattern observed in the human genomes, in the cancer genomes? So this allows us to propose more signatures. And again, going back to this groundbreaking work of Alexander et al., what they did is they utilized non-negative matrix factorization to come up with the set of possible signatures. And basically, the idea is that we have and all the cancer samples. And then for every sample, we have all this 96 category and the number of mutations in every category. And starting with this matrix, we decompose it as a product of those signatures that are being inferred and exposure, which is basically we represent this mutational matrix as a linear combination of signatures. With that, and that method has yielded to a catalog of mutational signatures that I represented here. Those mutational signatures are going to be referred, I will refer here as cosmic signatures. For many of those signatures are been quite well understood and been linked to respective mutagenic processes such as the UV light, but most of them are really there's very little understanding about most of them. So how we are going to, the next step would be to understand what those mutational signatures represent, what sorts of molecular processes or environmental processes are being cause emergence of the given signatures. Why do we care about it? Of course, we do care because we'd like to understand the mutagenic processes, the processes that eventually lead to cancer. It's also important because it helps both diagnostic and prevention. If we understand that certain environmental factors are responsible for particular signatures, then it helps us to identify those mutagenic factors and avoid them. And equally importantly is the therapy. So, as I mentioned in the first slide, we have the mutational, the deficiency of certain DNA repair pathways can be direct causes of appearance of mutation signatures. And, our understanding of which pathway is really deficient may help us to identify complementary pathways that need to be attacked with drugs in order to kill cancer cells through the synthetic lethality process. So how we can start to understanding those signatures using computational methods. So, well, we have to appreciate that, those signatures may be caused by germinal mutations, can be caused by environment, but also that there is a relationship between mutational signature and the gene expression network and pathways. And understanding of those relations will help us to understand the mutagenic processes or other molecular processes that may be responsible or otherwise associated with those signatures. Okay, so in my group, we've been working for a couple of years on those, trying to understand processes. And today I will focus on the approaches that are related to gene expression and mutation gene networks, because those are two questions that are most closely related to network biology. Obviously, one of the natural question would be, are there any genes whose expression or pathway whose expression correlate with emergence of a given signature? So that's the first question we ask. And for this question, we simply develop a very simple approach, assuming that we have a set of cancer patients for which we do have both mutations and also gene expression. We can then consider for every patient, we know the expression of all the genes within given cancer tissue, and also for every patient, we can also infer the strength of the signatures that are being observed in a given cancer type. So this allows us to consider every pair of gene and signatures and ask the question whether this gene expression of a given gene is correlated with the expression of with the strength of a given signatures. Many of those correlations will be non-significant, so we dropped. But the remaining significant correlations will lead to the signatures by gene expression matrix that we would like to explore more deeply. In order to explore this matrix, we clustered the genes by their mutational patterns, by their association with mutational signatures. So having clustered this, then we can dive into individual cluster and ask the question: what are the pathways? What are the pathways that are correlated? That are what are the genes which and pathways which are in each of those clusters? As an example, we analyze breast cancer data in this way, and for trying to understand what are the molecular underpinning of the mutational signatures that are observed in breast cancer. So in bottom here, you have these numbers of the mutational signature, but at the top, I just described what is known about the signature. So two signatures that are clock related, namely that are observed to be associated with patient age. There's the so-called ApoB signature that is often associated with immune response. This homologous recombination deficiency signature that we know is associated often with breast cancer. So just from this this clustering, we can gain at least understanding about those two clock-like signatures, and we can appreciate that one of the signatures is associated with cell cycles, and the second signature is not. Instead, it's being associated with a repair pathways, which is nuclear excision repair pathway, NER pathway, which is something that was not so clear without this type of analysis. There's a signature 8 that we don't know what it does, and because it's correlated with all the other signatures, we still cannot really understand it well. So this kind of analysis gives us some view of the mutational processes and they're associated with potentially DNA repair pathways and some insight into this process, but there are some limits. For example, this cluster here is highly correlated. So we'd like to have a deeper view into the relationships. So to have a bit, we need to leave just simple correlation and try to look at a correct relation to correct the relationship between any pair between say genes and signatures to see what is the relationship between gene and signatures if we correct for all the other confounding effect. So we decided to use it, and this is work by Arbata that recently joined the group. We decided to do it by constructing a network that will contain both signatures and gene expression, where both signature and gene expression have their activities. The activity of the signature is simply the what we call the exposure, the linear coefficient for a given patient, and the activity of gene expression would be simply expression of the gene of given patients. And then we would like to construct the graph not by simply correlation coefficient, but by using partial correlation coefficient and a sparsification procedure. So basically, for every node, now we forget that these nodes are different. They all have activity. So for every node, we would like to explain this activity as a linear combination of some sparse set of other nodes. So this will provide us with a directed graph where where for each node we will consider all the other nodes whose activity was relevant to explain activity of a given node. This some of the edges would be a bit directional, so that we won't be able that both the activity of this node requires activity of the other node. So some of the edges will be bidirectional, some will be unidirectional. We have some additional procedure to reduce the number of bidirectional edges, but at the end of the day, perhaps the most important part of this approach is the sparsification. The direction is perhaps less important. With that, we've been able to construct, gain much deeper understanding of the of the same breast cancer data. Now we can appreciate that that we have directed edges from double-stranded break repairs, which is homologous recombination differences to ApoBec. So we know which signature goes which one. We also can now appreciate that ApoBec signature is related to immune response, and we have the immune response regulatory T-cell activations associated with ApoBec, which makes biological sense, and the cryptic signature 8 that was not clear what it's associated with is now being suggested to be associated also with nerve pathway, and there are some experimental evidence that indeed might be the case. So indeed, by sparsification the network using the network-based approach, we have been able to dive much deeper into this understanding which signatures are associated with which processes, molecular processes, in breast cancer. We'll repeat this analysis to other cancer, but I am not going to discuss it further. So the second, so far we looked at how those signatures may be associated with gene expression and whether we've been able to pick up some of the more broader signals like dysregulated pathways that may explain those signatures. Equally, we can ask whether there are some mutations, some cancer driver genes that might be also behind some of the signatures. And motivation why that is expected to be the case is, for example, we know that mutation, germline or copy number variation in BRCA1 leads to homologous recombination signatures, which is known as signature 3. There was also observed that mutation with ARCC2 gene, which is one of the genes of the nerve pathway, leads to signature 5. So with that, we will expect that there is mutations, some of the driver mutation, especially if those driver mutations are hitting DNA repair pathways, those are likely to be also somehow associated with specific signatures. So in order to, again, linking back to network-based approaches, we ask the questions whether some signatures are associated with some mutated subnetwork in cancer. To do so, we think of a mutational signature as a continuous trait. So we know that with every patient we have, with whom, in whose genome we have observed a given signature, we also have the strength of the signature. So the strength of the signature is being considered as a continuous trait. So we ask the question now whether we can identify mutated subnetwork associated with continuous trait. So this is something that my group has been considering in the context of also drug response. So the method will be the same. So I'll briefly explain the method without going too much into the detail. So we assume that we have continuous traits, say drug response, or in the case of this application that I'm discussing will be mutational signatures. And at the same time, we also have a set of patients of this response has been measured, and those patients or cells have also we have the information about which genes are mutated. And then also, in addition, we have a protein-protein interaction network on which we can overlay those mutated genes. So we basically, the idea is to identify the subnetworks that are associated with the given continuous trait without, again, going too deeply into the method itself. I just mentioned that we formulated this approach as optimization problem using integer linear programming, and for and the technique that we use is connected set cover. So we try to find the minimal set of optimal connected subnetwork that explain the continuous trait in the optimal way. So the rest of the slide here shows what we did with this approach in terms of looking at the drug targets. However, here is some results that we did obtain from looking using this method to study mutation signature. So here we have all the patients being sorted with respect to the strength of mutational signature in all these orange bars, and then below are the to the left are the subnetworks that we found being associated with a given signature, and the bars show in which patients given gene is being mutated. Just as going to the best understood signature out of all of them is homologous recombination deficiency signature, which, as we know, is associated with mutation in BRCA1, BRCA2, but also we can see that's also associated with TP53. There is also we have also ApoBec signatures that we see this associated again with TP53 and also notch signaling pathway. And I haven't pointed it when I showed the gene expression analysis, but in these networks we also saw the negative correlation between the signature and notch activity. So we kind of could in this way associated some the mutated genes with pathways with signatures, and this information is relatively consistent with also what we observe through the gene expression analysis. So this is summarizing a little bit other challenges that are related to analysis of the mutation signature. I would like to just mention other computation challenges that are not necessarily related to networks, however, provide an interesting computational questions. So I started with this slide that explains that the final results of the mutations that we observe is an interplay between DNA damage and DNA repair. And at the same time, the methods that infer mutational signatures are the most popular one, at least, and actually most popular ones are relying on the decomposing of the patient mutational profiles using additive model representing the that those mutational signatures, that the mutation process are independent. This is not the case. Certainly, different DNA repair deficiency can be superimposed with different DNA damage deficiency, and we can obtain a large number of signatures. And in fact, this is what we observe. So, for example, here we have two mutational signatures that are very different that are associated with mismatch repair. Why? So our current understanding is that although those the mismatch repair deficiency is common for those patients, however, the causative, the mutational processes that are primary, that the damage processes are different. So for that, to deal with that, we have to study models where, rather than using non-negative matrix factorization, we accommodate the fact that we have those two processes, DNA damage and DNA repair, that occur not independently, but one acts on the other. So the DNA damage is still represented as a linear process, but the DNA repair is now we model as a process that acts on those primary DNA damage step. Other challenges is that DNA repair processes do not act uniformly on all whole genome, and also DNA damage processes do not act uniformly on the whole genome. For example, there are some DNA repair processes that are really focused on that DNA repair machine is attracted to the actively transcribed genes. Also, some processes like ApoBac processes is really giving mutation in the single-stranded DNA. So this is again related to specific processes that create single-stranded DNA. So the things is generally to summarize that things is really much more complicated than just assuming that the distribution is uniform, and some computational method and approaches to model this dependency have been developed, including some of the from our group. Okay, and this was I'm still not sure if you guys could hear me. I hope you could. But let me summarize here. I would like to thank all the members of my group and also collaborators, funding from NCBI, and just to mention that we have also the open positions, postdoctoral positions in the group. So thank you all, and I will try to see if I can, close this presentation. Thank you.